about your work.
So my work is mainly focusing on deeper understanding of a CatSper channel, which is a sperm-specific calcium channel. And then I haven't really thought about it working translational aspect of that channel in the beginning without any background on that aspect. But then it's kind of funny that when I recall, actually I went one of a very early MCI conference back in Seattle, like 2007 or 8, when my previous postdoctoral advisor was invited, but then he couldn't make it. Then I learned about those concepts of a male contraception, and as back at time, as a mother of having two young children, toddler and infant, I thought it's gonna be really cool to have those. But then it was forgotten in the middle during my postdoc. Then when I become assistant professor, one of my postdoctoral fellow actually got a fellowship from MCI, which has been a huge impact in my lab, and also the way we think how it can really contribute, not just within the understanding, but kind of a contributing to a bigger impact on society. So while initially I was a very much basic scientist by training, but I'm learning a lot with that kind of a line of thoughts. And then in this field there are not so many actually fellowship for postdoctoral or graduate students in reproductive biology field. So I know the Male Contraceptive Initiative not only support especially trainee level, not only just with a goal to male contraception, but more supporting the young people in the field in general. So I think it was really good momentum for not only my lab, but my trainee and my line of thought. Now at least I think about those translational aspects at the corner of the brain all the time. So it's good.
Good.
Yeah.
And tell me about the relevance of CatSper to male contraception.
Mm-hmm. So to be a male contraceptive target, I think it should be really male specifically expressed. So CatSper certainly one of the best well characterized male contraceptive target, because it is not only expressed only in the sperm cells and on the surface, it is also expressed post-meiotically. So when, of course, there is always pro and cons, but it's not any a drug, potential future drug, specifically targeting CatSper's one disrupt a spermatogenesis or sperm generation function, and then it can be on demand, or also because of it is so male specific. Actually, it has a double side coins aspect. Even if developed as a female contraceptive, in that case we wanna obviously develop as a non-hormonal contraceptive, because hormonal contraceptive for females already available. Then female doesn't have any sperm cells. As long as we can make sure the specificity, it's also gonna have a minimal or no side effect on the female side. So I think target concept wise, different from the practicality of how to achieve it, I think it's gonna be a very good contraceptive target, and people has been eyeing on it for a long time, but there were some hurdles in history why it was so hard to get it there yet. But I think
can you talk about some of those hurdles?
Yes. So now the structure at atomic level and in cell higher order levels are available, a one atomic level in 2021, and then in cell structure from my group in collaboration with Daniela Nicastro available, and it turned out to be, now it is much more understandable why it was so hard, because each complex channel complex, monomeric channel complex itself is made up of more than 14 like subunits. And maybe there is a possibility 14 is not the last one. So building all those 14 in the right position all together and then trafficking to the cell surface seems like either requires very specialized trafficking machinery or specialized or other germ cell specific system to build and target. So I think that was the reason. So because most of other ion channel, even if there are some other ancillary or auxiliary subunits available, they are formed complex together. The pore forming subunit itself, if they express in heterologous system, although with a slightly different biophysical property, they're still able to go to that cell surface. So that's why the in vitro, like a high content, high throughput screening was not possible. And of course, there are several excellent groups who were able to directly use human sperm for screening, and they, to my understanding, they already got several good hits or lead compounds. But then what those chemicals can benefit still from in vitro screening, if it can be achieved, is while we see the signal, we don't know the binding site if you are only using the native cell. And so there was kind of a hurdle, kind of a slowdown. I cannot say it is impossible, especially now with the atomic structure. Maybe they can do like a homology modeling and other better in silico screening, but it was really hard to know whether when you see a signal, for example, calcium imaging or like a flipper screening, when you see a positive signal, you wouldn't know it is the signal went up indirectly or directly bound to the CatSper. And then when you don't know the exact binding site, then it's going to be hard to know the mechanism of action and then screening out against others. So I think reaching surface expression and getting heterologous expression screening platform was the main hurdle.
Mm-hmm. Can you talk a little bit about what MCI's support is doing to help advance CatSper as a contraceptive target and what it means for you and your lab?
Yes. So as a small, like a junior faculty ran lab, sometimes when you think you had a good idea, it is really hard to launch like an unfunded project for a long term or seriously. But the MCI's like a scholar kind of award, it's not long term, it's not huge investment, but good amount of money. You can test some idea with so the SOCAR award I got awarded and received from MCI really helped to test those kind of tingling idea, but was not so sure whether I can do it or not. So when we got the idea of making like a chimeric channel, which is actually kind of a common approach in ion channel field, because ion channels are made of a very well conserved domain, like for example voltage sensing domain versus pore domain. So domain swap or protein engineering concept is very common in ion channel field. So I thought when I found a paper back in 2019 from a UK group reporting like an ancient, not exactly ancient, it's like a diatom eukaryotic ortholog of CatSper, I was very pleased because we knew bacteria like an ancient homolog, ortholog of the CatSper, which is Natchback. But then, bacterial channel is too far away from, mammalian channel, CatSper. But this diatom channel, which is at least eukaryotic, and then the group reported the channel itself can go to the surface. So we thought, okay, what's gonna happen if we try do domain swap so that the well-known, notorious like ER-Golgi trapping of the CatSper channel can be relieved from this domain swapping and then serial effort of making deletion mutant, etc. So that idea fortunately got selected by the MCI, and then we were able to kind of test that idea, and then we are seeing some promising, still preliminary promising results. So, and with those results, that became kind of foundation for we can think bigger and then try like a bigger, like a NIH kind of fund. So it was really a starting point for we as a basic science lab can try something a little bit outside of our own comfort zone. Yeah.
Mm-hmm. That's great. I think that's one of our goals as a research organization is to promote new ideas, to promote things that may be considered risky by some, but have the potential to really change a field or potential to bring new ideas to the forefront. Mm-hmm. So it's just to say that I'm really glad we were able to support you.
Yeah.
Awesome.
Yeah.
Well, thank
really
you for
appreciate
your
it.
Of
yeah.
course. Thank you for your time. I appreciate this.
Thank you.
Yeah. Awesome.
Thank you for your support.
Mm-hmm.